Hello, everyone. I'm just going to wait a minute for people to join. Thank you so much for being here. All right. I'm Lauren Goldstein. I'm the Assistant Dean for Research at the School of Public Health, and we're happy you're here at the latest in Public Health Research series. We're really pleased to have with us Professor Veronica Miller today, and she's going to be talking about biomarker validation as surrogate endpoints for drug approval. She is the director of the Forum for Collaborative Research, which is based in DC, and she'll probably tell us more about that as well. So welcome, Veronica, and I will give you the mic.
Thank you so much, Lauren, for the introduction, and good to see everyone. Although I can't see you because you're on my other screen, but I know you're there, so that's good. So yeah, today's topic is biomarker validation as surrogate endpoints for drug approval. And I'm really glad that the School of Public Health has this series. I think it's a good for all of us to hear more about what each other is doing. So my objectives for this talk is to, first of all, introduce the Forum for Collaborative Research. We're based in Washington, D.C., so I spend most of my time here, and so don't get as many opportunities to interact with our Berkeley campus colleagues. And then basically talk about biomarkers and their contribution to drug development, and then go specifically into one of our projects, which we call our Liver Forum NIT, that stands Non-Invasive Tests project, which is sort of where the research is happening. And I hope that with all of this, I can generate more enthusiasm. I know there is quite a bit for the process of drug development and its importance in public health. So the Forum for Collaborative Research is an umbrella organization in the School of Public Health, as I said, based in Washington, D.C. We work on several disease areas, including infectious diseases such as HIV, hepatitis B, transplantation-associated viral infections. Today, I will focus mostly on our Liver Forum, which looks at a chronic disease called metabolic dysfunction-associated steatohepatitis. And it's a chronic liver disease that affects, we're calling it the next public health and global health tsunami. It's associated with diabetes, type 2 diabetes, obesity, and it's really highly prevalent, especially the more progressed form of MASH, which is the steatohepatitis. MASLD stands for steatotic liver disease. So steatosis basically means fat. So you have fat in the liver, and that can cause hepatitis, which can lead to cirrhosis, etc. And there's a progression there. We'll go more into that later. So this is just using the Liver Forum as an example of how our forum projects are set up. We work with regulatory agencies such as the U.S. Food and Drug Administration, the European Medicines Agency, and the related European national authorities. Also with more and more with authorities in Africa, especially for infectious diseases. We, of course, work with academic clinical researchers, the patients, the industry. We work with the scientists in the industry. They know how to develop drugs and diagnostics and vaccines. And so we are very pleased to have them so actively involved in bringing their expertise in drug development to the table. And anyone else, there's foundations, consortia, professional societies, etc., that we bring in as well. And a lot of our focus always is to facilitate drug development, make it more efficient so that patients have access to more efficacious and effective and safe drugs than if in the absence of collaboration, things tend to take longer. So with that, I will just jump right into biomarkers. By the way, Lauren, if people have questions, will you let me know if there's a hand raised? Because I
Yes,
can't see him.
yes, no problem. I'll let you know.
Good. Okay. So I think many of us have heard about biomarkers across our lives and biological scientists, but what exactly do we mean by a biomarker when we talk about drug development? And the best source I can take you all to is called BEST. So that's biomarkers and endpoints and standardization and other tools. And it's a collaboration between the US FDA and NIH to specifically and clearly define the terms that we use in drug development. So a biomarker is a defined characteristic that is measured as an indicator of normal biologic processes, pathogenic processes, or biological responses to an exposure or intervention, including a therapeutic intervention. Biomarkers may include molecular. So if you go to the doctor and the doctor measures your liver function tests, such as ALT or AST, or measures your hemoglobin A1C, that's measuring molecules in the blood circulating. Histologic is if somebody takes a biopsy and looks at that. Radiographic would be an X-ray or other kinds of imaging, or of course physiologic characteristics as such. For this topic here, what is important to remember that the biomarker itself is not a measure of how a patient, an individual person feels, functions, or survives. And that will become clear why I am emphasizing that very soon. So basically, it's something that we can measure that has been identified to be part of a normal or pathogenic process. And I just want to clarify here too that in this age of omics and constantly identifying new proteins or new protein segments or portions, um, or other types of markers. There's so much that we see every day and publication after publication. But in this context, when we talk about biomarker, it's not something, some new protein X that has been identified. It's really something that we know is part of this process of other normal biologic processes, such as how collagen is involved in the formation of fibrosis and wound healing, um, or how collagen can be involved in the process of fibrosis leading to cirrhosis, which is a bad outcome. So we're not referring to the thousands and thousands of newly identified molecular entities through all these different omics projects. So when we talk about regulatory decision making, if the regulators are going to say this drug is approved for the following indication, they require demonstration, convincing demonstration that the drug has a clinical benefit. I'm not going to go into side effects here, but the overall clinical benefit, of course, has to also bring in the potential downsides of a drug. So the overall clinical benefit, what the benefit to the patient is, what the drug does, needs to be larger than the potential risk of what damage the drug could do. So when we say clinical benefit, that assumes that the damage is not so bigger than the actual benefit itself. And the FDA defines clinical benefit as how a patient feels, functions, or survives. Now, any of you involved in clinical research on different diseases and different therapeutic interventions or vaccine interventions or even behavioral interventions, you know that these functions, how a patient feels, functions, and survives. Of course, survives is very clear. It's either yes or no, but feels and functions, those are very kind of vague concepts, and we need to really clarify what they mean. Um, and that can become a little bit tricky. So let's go into that a little bit more. So examples of how a patient feels, functions, or survives is obviously one is mortality. In the early days of HIV, we called it AIDS at that time. We don't use that anymore, acquired immunodeficiency syndrome. But if patients experience the new AIDS-defining events such as PCP or Kaposi sarcoma or tuberculosis or a cancer, that was a clinical event. Or, of course, if they died. If patients with liver disease have end-stage liver disease, or they need a liver transplant, or they have full decompensated cirrhosis, or they get a liver cancer, those are all clinical endpoints that, of course, determine how a patient feels, functions, or survives. And of course, we don't want patients to get to that stage. That's why demonstrating something, a benefit of a treatment before patients get to these stages is so important. One other thing to keep in mind is the kind of different, what we call the context of use. So are we developing a biomarker to be a diagnostic? Does the patient have, or the person, the individual, do they have the disease? Yes or no? Is it prognostic? Does it tell us how quickly the disease is likely to progress? Or is it predictive? Is it going to predict how likely that individual is to respond to treatment? PK/PD stands for pharmacokinetic, pharmacodynamic. That is, can we see how the drug actually engages the target? And depending on how much drug there is, the dosing and the distribution of the drug in the body, and its the pharmacodynamic effect, such as lowering viral load or lowering blood pressure, etc. So that's also a very important context of use. And then the one I want to focus on today is as a surrogate endpoint in a clinical trial. So going back to our friend, the BEST, it really is a very useful tool for anyone interested in these topics. So we learned before that what we really need to demonstrate is a clinical benefit. So we need to show that there's less people dying, there's less people getting liver cancer, there's less people needing a liver transplant, for example. But so that is what we eventually need to see, but we don't want to see have to follow the patients all the way to a liver transplant just to say that people in arm A did not need as many liver transplants as people in arm B, because that would be well, in some cases we still have to because we don't have any other biomarkers, but we don't want to be at that stage in drug development. So an endpoint is used in clinical trials. A surrogate endpoint is an endpoint that is used in clinical trials as a substitute for a direct measure of how a patient feels, functions, or survives. So we don't have to go all the way to the patient's liver no longer functions. We can predict that the liver will stop functioning at a certain period of time based on some biomarkers that we can measure. And a surrogate endpoint does not measure the clinical benefit of a primary interest in and of itself, but it predicts the clinical benefit or harm based on previous clinical trial data, pathophysiologic, or other scientific evidence. And maybe if I give you some examples that might clarify this, and please do ask questions if I'm going too fast. So in HIV/AIDS, when that disease was first identified, and eventually the virus, a human immunodeficiency virus, was identified as the virus that caused the disease AIDS. The initial trials were done based on how many AIDS-defining event the patients have and how many patients died. And with more understanding of the virus itself, the virus replication dynamics, our ability to use PCR to quantify virus, all of these different technological advances helped us to understand that what was driving the disease was the virus replication. And by measuring virus RNA, and the dynamics of HIV RNA, we had a very good idea of what was happening. And so the endpoint to prevent clinical progression to another AIDS-defining event or death was to suppress the virus so that we have no detectable HIV RNA in the circulation. And I'm sorry, there's a typo that should say plasma here. So if we have undetectable plasma HIV RNA, we know that the patient, first of all, it's very good because the patient is not going to transmit the virus to somebody else. And number two, that patient, as long as the virus remains suppressed, can live in nearly pretty much a normal lifespan. Another one in chronic kidney disease is we don't have to wait for the patients to have to go to dialysis because the estimated glomerular filtration rate, often called EGFR, or serum creatinine, can tell us how well the kidney is functioning. And so if we have a drug or treatment that improves the EGFR or improves the serum creatinine level, that drug can get approved for chronic kidney disease, and we don't have to wait to see if it actually leads to more kidney dialysis, because that relationship has been shown over and over again. And hepatitis B, again, we go to nucleic acid testing with undetectable serum HBV DNA, as well as hepatitis B surface antigen loss. HCV, I think many of you know about the sofosbuvir and other direct-acting antivirals that now makes HCV curable. If we have sustained virologic response for 4 weeks or 6 weeks or 8 weeks, we know that that patient is cured. And then finally, for type 1 or type 2 diabetes, we have the hemoglobin A1C. I've put an asterisk by some of these because either I myself previously, when I was working in Frankfurt, we worked on the HIV/AIDS plasma RNA and contributed to that approval, and also redefining how drug resistance plays into this. And then with the forum, we of course continue working on HIV in terms of HIV prevention trials, but the forum has worked on hepatitis C and is currently working on hepatitis B in terms of defining the biomarkers that will predict curative treatment. So why are these important? Because when we have biomarkers like the ones I mentioned, for example, HIV RNA or in hepatitis, if we can demonstrate complete loss of hepatitis B surface antigen, that's a protein, or DNA, and there are many other biomarkers we're looking for now. We're looking at in hepatitis B clinical research. We can accelerate drug development, and we've shown that over and over again. So we don't have to wait for the clinical event to happen. If that weren't the case, then those direct-acting antivirals that cure hepatitis C, you would have had to do those studies and then again compare how many patients went all the way to cirrhosis, liver cancer, and death before you could confirm the clinical benefit. So we accelerate drug development. That's a benefit to everyone. And we can also treat patients and improve their clinical condition before they have irreversible clinical damage. Yes, for example, the liver is very resilient and can regenerate, But there comes a point where too much damage is too much damage. And so we can reduce morbidity and mortality significantly if we start treating based on biomarker evidence. And as I hope you will also see with the liver disease case that I'm going to present, we can improve the scientific rigor because we have often a much wider range of numbers to work with and precision as well. Instead of saying, of course, you know, again, death is a very precise outcome, which we want to prevent. But even progression to cirrhosis is a very well, you know, you can diagnose the progression to cirrhosis. But when we have biomarkers where we have several logs of values, we have a much bigger range and often much more precision. Also, to then start thinking about dividing patients into subpopulations as appropriate. So it just really brings a whole different science to the game. And I just wanted to bring one example that a colleague of ours presented at the last Liver Forum meeting. She's one of the directors at the Foundation for NIH, which works with pharmaceutical companies on different disease area. And she presented the case that FNIH had sponsored on bone marrow density as a regulatory endpoint. And I'll just show a few of the slides here. This is a very interesting case. So this is as measured by DEXA. You measure bone marrow density, and it's now been accepted as a surrogate endpoint for fracture and osteoporosis clinical trials. And again, you don't really want patients to get fractures before you declare that the drug didn't work or that the drug did work. So in this case, they were able to show, based on a lot of individual patient data from many different trials, that the percentage change in bone density in 2 years is enough to predict a future fracture. So if your bone marrow density goes down, this can reduce the trial size for demonstrating new drugs that work in bone marrow, in bone density and osteoporosis. So instead of needing 10,000 patients, you can see the difference and you can declare efficacy based on just 1,000 patients. And you can do it in 2 years instead of 10 years. So you're really reducing the cost of the drug development here. And that's really so important. And also the regulatory confidence in the biomarker endpoints, because again, of the range and the precision of the values that you can generate. So here, some of the key lessons were that, you know, for many of these kinds of studies, we need pooled patient level meta-analysis and, you know, to look at things in different statistical modeling and different ways of looking at the data and cross-validation, etc. So this was just a very nice example of a publicly, well, FNIH-funded study recently in bone marrow. I keep saying bone marrow, I mean bone density related osteoporosis. And then I wanted to talk a little bit about uncertainty and certainty, because so much of what we do in clinical research carries with it a certain amount of uncertainty. And regulators are willing to live with uncertainty, more uncertainty, and very severe disease cases, and some of the very rare and very rapidly progressing neurodegenerative cases. Or, for example, when something like COVID came along, if you all remember, there were things called you know, they were approved initially as an emergency approval, an emergency procedure, which is the one that really allows the most uncertainty, because the the consequences of COVID-19 were so severe before we had vaccines and drugs to treat it. But outside of that emergency use authorization, when we talk about biomarker endpoints, first of all, we start with a candidate. What would be a good candidate? And then hopefully we can get that candidate, if it is a good candidate, to become what we call a reasonably likely surrogate endpoint, often abbreviated as RLSE. And it's reasonably likely to predict clinical benefit. And then once there's less uncertainty, more certainty, and this is through clinical trials and analysis of larger data sets, it can become a validated surrogate endpoint. And this difference is important because if you are demonstrating clinical benefit, like the first drugs for HIV, which depended on less disease progression and less death, you get full approval. And that is dependent on you're demonstrating that the patient population had significant improvements on how they felt, functioned, and survives. Then we have a surrogate endpoint, which can lead to accelerated approval. And that is reasonably likely to predict clinical benefit. And that gets you accelerated approval. But regulators still need to confirm that clinical benefit. So through data analysis of clinical trials where these surrogate endpoints are leading to accelerated approval, that relationship can be substantiated and become much more certain than it was at the surrogate, at the reasonably likely surrogate endpoint. So once that endpoint becomes validated, that endpoint, such as HIV, undetectable HIV RNA, or 4 weeks sustained virologic suppression and HCV, gets you full approval of the drug. But this is the crucial part here where people need to collaborate to be able to really substantiate that surrogate endpoint of clinical benefit. So that is what really brings a lot of challenges to the field. So now to go to our main case that I wanted to discuss today is what we're doing in the liver forum. And remember, this is just one of our forums. So this is the metabolic dysfunction associated steatotic, sorry, a spelling error there again, liver disease or MASLD, or a steatohepatitis, which is MASH. So hepatitis is worse than steatotic liver disease. Hepatitis, the itis part here means that there's a significant amount of inflammation going on. It's a very slowly progressing chronic liver disease, which means that from the time you identify as somebody as having too much fat in the liver and beginning of fibrosis, to the time when they would have an endpoint, that might be 10, 15 years. And we all know that those studies running for 10 or 15 years are impossible to do. So the current paradigm has the accelerated approval, which means that the drug can be marketed based on a liver biopsy histological assessment. That is dependent on the NASH activity score. I put that in quotation marks because it used to be called non-alcoholic steatohepatitis. Now we call it metabolic dysfunction associated, so it's MASH. But the name NASH activity score still stands. And so that's basically your NAS. And then fibrosis. And note that fibrosis is staged based on the biopsy between F0, that's healthy, and F4 means that you have cirrhosis. So it's a very narrow scale, and the same with the NASH activity score. 2 drugs have been approved based on this accelerated approval. One is Resdifra or resmetirom from Madrigal, and the other one is semaglutide Wegovy, which has now added MASH, MASLD and MASH to its indication. It was originally approved for weight loss. So the problem with the biopsy is that the traditional way, which is the approved pathway for FDA approval, depends on a liver biopsy where you go in with a certain size needle, and there's some help with ultrasound as to where exactly the needle goes. But the liver is not just like a uniform, a uniform consistency. There's all kinds of little pockets of inflammation and fibrosis and fat droplets, etc. And one of the key identifiers for using the biopsy for MASH is something called a ballooned hepatocyte. Notice that sometimes, people just call something by what it looks like. So it's a cell that's ballooned out. There's going to be fat droplets, there's going to be areas of fibrosis, etc. So in this study by Brunt et al., they had 9 different pathologists, very well-regarded liver pathologists, who scored this one biopsy. And they all identified, the different colors are the different pathologists here. And there was one balloon cell that was identified by all 9. Only one amongst the 19 or so different ballooning cells in this section. Only one was identified by all. And this really illustrates the variability in the reads. And there's many other issues with biopsy as well, especially because it really depends on where exactly the needle goes and what you pull out. So pre-treatment and post-treatment, if you're in different areas of the liver or just even small differences, you may see a completely different picture. So we do have AI, so digital pathology, assisted with machine learning and artificial intelligence. So here again, you see a normal liver section. And so this one is the so this one is with the Mason trichome stained section here, and the identical section without the stain, but applying, this is one of the companies that works in it. The company's name is HistoIndex, and they have a process called second harmonic generation. And it has to do with how the photons, the signals, light up the fibrotic areas. Now you can see fibrosis here, experienced pathologists would pick it up, but see how much easier it is to pick up here. And obviously this can lead to much better quantitation and characterization, because it's also important where exactly the fibrosis is taking place. There are other companies as well. I just picked out this one because I had this picture handy here. So this would obviously help a lot to already quantify and have a bigger range of the amount of fibrosis. And these companies are working on that. But in the meantime, we're stuck with the old method. And this is the phase 3 study that was published last year that led to the approval of resmetirom in NASH with liver fibrosis. And you can see here, so in terms of the percentage of patients who had NASH resolution, so less fat, less metabolic activity in the liver, and inflammation and ballooning with no worsening of fibrosis. But that scale is just really a couple of integers, right? Integers. It's not a big scale. But nevertheless, they were able to show that patients on the active treatment, a higher number of patients responded compared to the placebo. And the same as with fibrosis improvement. So the outcome here we're looking for is fibrosis improvement by 1 or more stages with no worsening of NAFLD. So if you include patients that are already at F2, because F0 means there's no fibrosis, you wouldn't want to include them. There were some F1 patients, but the majority were F2. And you can really go to F3 or F4, and F4 means cirrhosis, which you're unlikely to do in the follow-up time of the study. So you really did not have a lot of opportunity to show either improvement or non-improvement. And of course, they've gone back and looked at other biomarkers, including with histoindex and looking at the specific quantitation of fibrosis. But I just want to show you this to really say this is 2024 with all the technology we have, and we're basically using a scale of 0 to 4 to evaluate the effect of a drug on a very, very common public health issue. So we at the forum then put together a proposal, which we will work with the US FDA and the European Medicines Authority, representing all of our partners in the Liver Forum. And this is just a little bit of introduction again. You have fat in the liver, steatosis, and that leads to MASH, which is the more severe form. And this can kind of go circle back and forth. You exercise a little bit more, you drink less alcohol, although this is not supposed to be alcohol dependent, this is metabolically dependent, but you eat healthier, eat more fiber, more fruit and vegetables, etc. This can kind of cycle back and forth. But once you get into early fibrosis, again, you can kind of continue all the way to cirrhosis, and you can also regress. But this is a little bit more, not as frequent as just this sort of cycling back and forth. So you have increasing hepatic fibrosis deposition that leads to cirrhosis, which leads to liver mortality and liver-related outcomes and death. And so this is the clinically meaningful outcome that we want to avoid. We want to reduce liver-related outcomes and death. And at some point, we will have what we think of as a generally accepted or a validated surrogate endpoint. But right now, we're working on what is a reasonably likely surrogate endpoint to predict benefit based on those outcomes. So based on the rationale for how we choose a surrogate, there's many different papers that really discuss all the criteria that go into this. But let's just then come to this kind of principle here that the change in hepatic fibrosis should be the basis for an NIT, non-invasive test, based reasonably likely surrogate endpoints. So hepatic fibrosis leads to liver stiffness. So there are different ways of measuring the liver stiffness with different imaging or other types of ultrasound in FibroScan. For example, FibroScan is one of the ways to demonstrate that. And the other one is to look at it in terms of biomarkers, collagen-based biomarkers that are circulating in the blood. So we can either look at something that's physical, like a liver stiffness, or a collagen dependent. And of course, the change in liver fat content is also important and may provide some additional supportive evidence, but it's not the fat that kills you, it's the fat leading to fibrosis and cirrhosis. The fat inflames the liver to progress to end-stage liver disease. So we have elastography, from the Greek elasto, meaning the elastic, that kind of. So, for example, we can measure it with VCTE, which is an instrument called a FibroScan, or MRE, which is a form of MRI imaging that looks at liver elastography instead of liver fat. And ideally, we could say we want to change a biomarker 1 or change a biomarker 2. Of course, ideally, a patient would see improvement in both. But these biomarkers have their issues, and sometimes, depending on the physiology of the patient or on whether they have just eaten or the age of the patient, for example, in kids and teenagers, these collagen markers are different, behave differently because of the bone turnover. So there's different reasons why one biomarker or the other might work best. Of course, the highest certainty you would have if a patient improved in both. But that's basically our proposal, that we should, instead of biopsy, we should look at liver stiffness biomarkers. And this is from one of the papers we're putting together. It's almost ready for final review and for submission. So we want to go from the surrogate endpoint, which requires 2 biopsies. And if anyone has any illusions about this, it is a big little needle that goes into your side between the ribs and takes out a bit of liver. It's not a pleasant experience and does carry some risk with it. To kind of going into a non-invasive test, that's why the non-invasive is so important here. That would predict clinical benefit. So what we did is we basically went, because the data to support this can come from clinical trials, it can come from epi studies or other published studies. So we basically went and looked at all the studies available on VCTE, which is the liver stiffness. ELF is one way to look at the collagen level. It's called Enhanced Liver Function Test. And you can see we have a total of over 70,000 patients that were involved in these studies. So it's quite a big database. These are not individual patient data sets, but for some of these studies, we can go back and work with the authors of the papers to do some reanalysis and possibly collaboration on individual patient data sets. But anyway, so I cannot give you the complete results because that would take 2 hours. It was quite in-depth analysis, but basically this is the very top level. So VCTE for prognosticating disease progression came out very strong. And for documenting treatment change also was pretty good. MRE, not as good, sorry. I'm hearing somebody speak, but it's simply not because MRE is not a good test. That's actually a very good test, but it's more expensive, not as widely available, and we have less data on that. And again, ELF, a prognostic and treatment change is pretty good. So we have more work to do. But basically, what you can see is if we have an NIT non-invasive test get to accelerated approval, that gives you a drug on the market a lot sooner than if you have to wait all the way all the way to the end to demonstrate that you have a decreased progression to clinically significant liver events. And of course, decompensated cirrhosis is the way. So it really is a very important path that we're going down because, like I said, it's impossible to do these studies that long. Now, I can tell that I'm going to be running out of time. Of course, there's so many, and we've already engaged some of the Berkeley faculty, like Alejandro, to talk to us about, continuous versus categorical variables or cutoffs, and how do we interpret that the curve of the clinical event reduction and the NIT, et cetera. So there's still a lot of work to be done here, but we've made great progress. And so now, of course, the challenge is that once you have these drugs on the market, based on accelerated approval, as I indicated before, we now are obligated, or the companies are obligated to demonstrate the clinical confirmation. So now they need to show that if you follow these patients long enough, there will actually be reduced clinical events. And that means studies that are 5 years or longer, and some of them are going on. And the regulatory requirement, of course, is that you continue the placebo assignment. So then we get into this whole question of what's the scientific validity and what is the ethical validity. If the drugs are available on the market, those patients on placebo who progress faster are going to drop out of the study and get the drug, right? So you're losing that protection that the placebo gives you to protect to prevent bias. And also, you know, you have to try and keep the patients in the study for the scientific validity, but is that really ethical? Does the clinical investigator have equipoise? So there's, again, a lot of work to be done here in terms of ethics and scientific validity. So lots of opportunities for collaboration across the school. And I will just finish with the our some of the potential solutions we're working on. This is really what's I'm most excited with about the forum. What we're doing right now is to look at how can data collaborations help. And I will focus right now on reducing the placebo arm burden, which requires placebo arm data, which we can then use as an external or supplemental external control arm, and use that to also build synthetic cohorts. And a lot of work going on in this area. And regulators are very interested in hearing more about this. We do have a project called the MASH placebo arm database, and it's our lead data scientist is Margot Yan. Richard Halbrick is one of our consultants. She presented this at the recent Liver Forum. It's all on our website. I will just go through this very quickly. So we have received, or data transfer is in progress for about 2,000 patients. There's more to come. Data use agreements, data transfer and use agreements takes a long time. And but we're happy, we're very pleased with how far we've come. And we have now we have to actually get to the work of how do we harmonize these, because we want to use this as an integrated data set. So a lot of what Margot and her team have been doing is working on the CDISC-based harmonization of these data sets. And I won't have time to go into all of this. Margot is on the call, and I think I will suggest that next time she talks just about this. But it's an incredible amount of very detailed, very repetitive kind of work to do this data harmonization across the different trials, because as you can imagine, there's lots of areas, even if these are all meant to be submitted to the FDA, there's still a lot of different data formats, etc., and the kinds of, you know, the time sequence and etc., etc. So a lot of harmonization needs to be done. I think one of the good examples here is how she's been working in terms of how many domains. This is just based on two studies, the ENYO study and what used to be Sharing Plow, which is now Mirum has that data. So the domains that both trials included, this is just based on two, is 16, although you have a total of 35 domains in our harmonized data set. And so when the third trial comes in here, that will all be repeated. So it's really an iterative process of data sets. It's not your traditional learning data set that then gets applied to a confirmatory data set. This is really with each new data sets that come in, this process and this pipeline will be repeated. So that's something that she has set up and demonstrated that it can be done. And like I said, I'm just going to go through all of this, just talk about some of the analysis we do. Of course, the placebo response, talking about both the biopsy, the regular notes from the pathologist. For some of these data sets, we'll also have the digital pathology as well. So we can really look at in the placebo arm, how much placebo response do we see, both in terms of the histology as well as the NITs. So the ELFs and the other liver function tests and the liver stiffness, etc. And so then we also want to work on how can we use this cohort to really generate synthetic cohorts. And one of those methods is to use a digital twin methodology. And again, I know there are people very much interested in all of this. And at this Berkeley School, we've already talked with Alejandra about this. And we've also talked with the Center for Targeted Learning on using this data set for causal inference applications, etc. So we're very much open to collaborating across the school with PhD students and investigators and fellow, people who are really interested in developing. We have so many statistical issues to work through, and in terms of how we look at the data, etc. So it's hopefully become a very interesting tool for everybody in the school. Just
Veronica,
wanted,
you're just about there. We're just about out of time, but thank you so much.
yeah.
I'll let you, finish your slide.
Right, wanted to, the key people, of course, everyone has been involved here, but the key people here that have been advising the program directly, of course, Margot Yan is, like I said, the director of the Data and Analysis Center. Edith Dwyer and Kathy Ann work as research associates in our office here, and of course, all the companies we work with.
Fantastic. Thank you so much. We really, really appreciate your time and sharing your amazing work. It's a great, great talk. I know that people probably need to go, but if anyone did want to ask a question, there was one question that came in. Why is eGFR considered a surrogate endpoint when it's a direct measure of kidney function?
Right, that's very good. You picked it up. It's a kidney function, and it is a kidney function, but it's not a direct measurement of kidney of the clinical event, which is the need for dialysis, kidney failure, basically. So kidney failure is the clinical benefit, the clinical event you're trying to avoid, and your kidney function can predict that. But that needed to be demonstrated.
Got it. Got it. Thank you. Great. Well, I put your email in the chat for people if they have further questions. And also I put the link to the forum website in the chat.
Thank you.
Thank you so much again for sharing your work. And thanks everyone for joining. Hope you have a good rest of the day.
Good. Thank you.
Take care. Thank you.